Hello, my name is Jonathan Lynch, and the title of my talk is New Routes for Agriculture, Phenes, Phenotypes, and Phenotyping for Soil Resource Capture. Our goal is to develop tools to address three primary challenges facing global agriculture. The most important of these is drought, which is projected to worsen this century as a result of global climate change. The second is nitrogen. Intensive nitrogen fertilization in rich nations causes massive environmental pollution and is unsustainable, while in developing nations low nitrogen availability limits crop yields. And the third is low soil phosphorus availability, which is a primary constraint to life on Earth. Now I will talk about three phenes of interest. The first of these is multiceriate cortical sclerenchyma. Here is a cross-section of a normal maize root. Hannah Schneider discovered that some genotypes of maize and other cereals form multiceriate cortical sclerenchyma, shown here in the outer cortex as small cells with thick cell walls. The MCS lines are rich in lignin, as you can see here on the left. And that lignin is associated with greater tensile and root tip bending strength, as you can see here. MCS enhances penetration in hard soils. Maize and wheat lines with MCS had greater penetration through hard wax layers in mesocosms. In compacted soil in the field, maize lines with MCS had greater dry shoot biomass and greater root depth than non-MCS lines. MCS is under genetic control, as shown by this Manhattan plot. This is a gene of interest with a heritability of 0.64. The second phene I will discuss is xylem perforation plates. This is research of Chris Strock. Xylem perforation plates are found where two xylem vessels adjoin, as shown here in the longisection and cross-sectional views. Chris found substantial genotypic variation in the length of xylem vessels, as shown here, with the short vessel phenotype on the left and a long vessel phenotype on the right. Chris also observed substantial genotypic variation for the height of the perforation plates, as shown here, with the prominent plate on the left and a shallower one on the right. Hydrodynamic modeling indicates that the height of perforation plates affects water flow. You can see here in the top panel, tall plates reduce water velocity in the xylem lumen, whereas short plates do not. To determine the effect of xylem vessel length on water flow, Chris compared the theoretical conductance measured by applying the Hagen-Poiseuille equation to measurements of xylem vessel diameters from root cross-sections with actual water flux through root segments at known pressure head. And he found that vessel length was associated with the ability of root water flux to approach its theoretical capacity. In other words, short vessels restrict water flow. Under terminal drought in field rainout shelters, maize genotypes with high conductance xylem morphotypes had nearly 3 times the yield of related genotypes with low conductance xylem morphotype. This is an interesting phene for drought tolerance. The final phene I will present is parenchyma cell wall thickness. We have observed that anatomical phene states that reduce the metabolic cost of soil exploration improve rooting depth and yield under drought in maize. For example, root cortical parenchyma cuts root respiration in half, increases root depth in the field, and more than doubles yield under drought. As another example, reduced cortical cell file number cuts root respiration in half, increases root depth in the field, and doubles yield under drought. As a third example, large cortical cell size cuts root respiration in half, increases root depth in the field, and more than doubles yield under drought. This led Jagdeep Sidhu to hypothesize that cell wall thickness may be useful also. Indeed, Jagdeep observed large genotypic variation in maize for the thickness of parenchyma cell walls as shown here, with a thick-walled genotype on the left compared to a thin-walled genotype on the right. Using root slice, our geometric model of root anatomy, we can ask if the genotypic variation in cell wall thickness we have observed is likely to make a difference for root metabolic costs. Root slice predicts that thicker cell walls will reduce cytoplasmic volume in a root by 36% and increase cell wall content in a root segment by 65%. Root slice predicts that thicker cell walls will reduce mitochondrial density in a root segment by 35%, phosphorus content by 53%, and nitrogen content by 54%. The magnitude of these effects should be significant for soil exploration under resource limitation. Indeed, real plants with thicker cell walls have less root respiration per gram dry weight and also per centimeter of root length under drought and non-stress conditions. Maize lines with thick cell walls perform substantially better under drought stress than lines with thin cell walls in the field. So this is an interesting phene for drought adaptation. I now turn to the second part of my presentation about integrated phenotypes. My first example is the synergy of metaxylem anatomy and root architecture in Phaseolus. This research was led by Chris Strock. Phaseolus acutifolius tepary bean is adapted to drier environments than its close cousin Phaseolus vulgaris, the common bean. Acutifolius has root architectural phenotypes that make it deeper rooted than vulgaris, but both species show large range in xylem conductance, as shown here. Chris grew low conductance and high conductance morphotypes of both species under drought in the field in Arizona. In this environment, water was more available at depth, as you can see in the right panel. He found that in the deep-rooted acutifolius, greater xylem conductance was beneficial under drought stress, whereas in the shallow-rooted vulgaris, greater xylem conductance was harmful. This is a striking example of the importance of interactions between root architecture and root anatomy for drought adaptation. The second example I will present is integrated root phenotypes for drought tolerance in maize. This research is led by Stephanie Klein. Stephanie found that drought tolerance among 400 diverse maize lines in the field was related to clusters of integrated phenotypes. Single phenes did not explain variation in drought tolerance among the whole set. Clusters associated with improved drought tolerance consisted of phene states that enable greater soil exploration by reallocating internal resources to greater root construction. These include increased parenchyma content, larger cortical cells, and fewer cortical cell files. Restrict uptake of water to conserve soil moisture. These consist of reduced hydraulic conductance, narrow metaxylem vessels, and finally improve penetrability of hard, dry soils, such as phene states like thick roots with a larger proportion of steel and smaller distal cortical cells. We now know that smaller distal cortical cells is actually the MCS phenotype I showed earlier. The third example I will present is phene synergism for nitrogen efficiency in rice. This research is led by Ishan Ajmera. Ishan developed a rice implementation of open SymRoot. Here we see the reference phenotype IR64. Linking OpenSimRoot to the crop model ORIZA identified 8 root phenotypes with superior yield under low nitrogen supply than IR64. Here we see projected yield of these phenotypes under low nitrogen over the past 33 years and projected next 80 years of weather. This is IR64, and this is the performance of the better phenotypes. Some phenotypes had substantially greater yield than IR64, over 80% increase. Interestingly, the better performance of these phenotypes was due to phene synergism. Here we see the vegetative growth of the phenotypes under nitrogen stress compared to IR64, which is the purple line. Their expected performance based on the additive combination of their phene states is actually worse than IR64 in all but 2 cases, which is not surprising since IR64 is an exceptional cultivar. The reason they beat IR64 is because of synergism among phene states. In other words, greater than additive responses, as you can see here in green. These results and others show that the utility of a phene state is influenced by interactions with other phene states in an integrated phenotype. This is hard. For example, if each of 5 phenes of interest exists in one of 4 states, in other words, very steep, intermediate, and shallow nodal root growth angle, for example, there exist 4 to the 5th, or 1,024 integrated phenotypes, each of which should be evaluated in different environments. How can we evaluate so many phenotypes? One option is multi-objective evolutionary optimization to explore phene space. This research is led by Harini Rangarajan. Harini used multi-objective evolutionary optimization to evaluate 22 phenes, each in 5 states, resulting in 5 to the 22nd phenotypes, evaluated in 25 levels of nitrogen and phosphorus, resulting in a decision space of 6 times 10 to the 16th phenotypes. She co-optimized against 6 objectives, conducted 250,000 evaluations, resulting in 2,000 optimal solutions. Shown here in the left against a nitrogen biphosphorus fitness landscape. One conclusion was that co-optimal phenotypes may differ in how they meet performance objectives. For example, here we see phenotypes with differing biomass shown as the black pie slice, even though they are all co-optimal against all 6 objectives. Another result was that contrasting phenotypes could perform well. As shown here, these 3 phenotypes all perform well under low nitrogen, yet they have different individual phene states. It's their integrated phenotype that gives them an overall performance, not the individual phene states. Machine learning will enable us to do this faster. This research is led by Ernst Schaefer of the University of Nottingham. Ernst ran some simulations across a 17-dimensional parameter space, used machine learning to restrict the parameter space to the most favorable parts of the landscape, and repeated this iteratively. You can see here that with each wave he discovered maize root phenotypes with better growth under nutrient stress. That's wave 2, wave 3. After sampling just 1,000 phenotypes, Ernst was able to eliminate 98.3% of a 17-dimensional parameter space, therefore massively improving the efficiency of this process. This is a very promising approach. I will conclude by talking a bit about phenotyping, starting with phenotyping root architecture in the field with shovelomics. Shovelomics consists simply of manually excavating, rinsing, and imaging root crowns in the field at the rate of about 2 minutes per plot for a variety of different crop species. Alex Bush developed DIRT, digital imaging of root traits, capable of extracting multiple architectural metrics from two-dimensional images of the excavated crowns. As an example of how this works, crown root growth angle varies among maize lines from quite shallow to quite steep, as you can see here for the nested association mapping panel of 5,000 entries. This research is led by Hannah Schneider. After 4 years of phenotyping 480 maize lines in the field with shovelomics, we identified ZM-CIPK15 through GWAS as being associated with this phenotype. This gene is expressed in roots. Mutating this gene results in steeper crown root growth angle, as you can see here. Here's the data for root angle in the field under low nitrogen and drought, showing a steeper phenotype for the mutant. OpenSimRoot predicts that this change in angle should increase nitrogen capture and growth under low nitrogen conditions, which was confirmed in the field under low nitrogen conditions. This shows N15 capture under low nitrogen conditions was greater in the mutant, and the growth of the plants under low nitrogen was greater in the mutant. We are developing a next generation platform called Root Robot DIRT 3D. Root Robot is led by Xiaomeng Shi. This is a tractor-drawn robot that lifts, slices, and cleans maize root crowns without human assistance. Here's the mobile unit prototype in the field last year. The video shows a crown being processed and deposited in a tray for delivery to the imaging unit. The imaging chamber automatically inserts a crown and generates 3,600 images from it from all sides. Alex Bush team at the University of Georgia has developed a 3D version of DIRT capable of resolving many architectural features, even inside the crown, as shown here. Suxing Liu is the postdoc leading this effort, and here you see some of the traits being extracted from DIRT 3D. Root anatomy can be phenotyped with anatomics. Anatomics consists of laser ablation tomography, which we invented at Penn State. It's a pulsed UV laser ablates a sample as it is imaged, as you can see here, resulting in a 3D image stack with anatomical detail. This can even be done in intact soil, as shown here. Here's an example showing a field-grown maize root with root corticolerenchyma in green and mycorrhizal fungi in yellow, showing that the fungi do not inhabit Erenchyma lacunae. Here's another example showing nematode feeding sites in a field-grown barley root. Images are analyzed by root scan to extract anatomical phenotypes in 2 dimensions. As an example of how this works, using the same maize diversity panel I noted earlier, grown in the field, we collected root crowns via shovelomics, ablated them with the laser, analyzed the resulting images in root scan, and via GWAS found that ZM-BH-LH121 is associated with root corticolerenchyma formation. This gene is expressed in the root cortex. An overexpression line increased root corticolerenchyma formation under low nitrogen, drought, and control conditions, here in blue, while 2 RNAi lines had less RCA formation, here in green and red. A CRISPR line had less RCA formation under all 3 conditions, shown here in blue. A mu insertion line had less RCA formation under low nitrogen, shown in blue. These results show the value of the anatomics pipeline for phenotyping root anatomy in the field. We are developing a next generation anatomics. LAT 2.0, led by Michael Williams, includes sample automation that greatly accelerates processing time, Michael has also been able to integrate hyperspectral imaging with LAT so we can get 3D data regarding tissue composition. So here's a sample being imaged by hyperspectral camera on the left immediately after imaging with LAT on the right. Here, for example, we see that hyperspectral imaging can detect MCS, multiseriate cortical sclerenchyma. Here in the red region, in the outer cortex, the peak at 500 nanometers is much greater in the MCS line than the non-MCS line. We think this represents increased lignification in the MCS line. Ezen Moko Benson at the University of Nottingham is developing RootScan 3D with machine learning to detect anatomical phenotypes without human assistance. In fact, this is able to detect anatomical phenotypes better than humans. And here we see that RootScan 3D can detect shapes in 3D, in this case May's xylem vessels from image stacks. Finally, I would like to talk about a method to non-destructively measure root depth in the field, LEADER. This work is led by Molly Hanlon. Here's Molly with a handheld X-ray fluorescence spectrometer that, as you can see in this spectrum, can non-destructively measure the metal content of a plant leaf. LEADER stands for leaf elemental accumulation from deep roots. The concept is that some metals are more available in shallow soil horizons, others more at depth. In this case, both a shallow and a deep-rooted maize line can accumulate the blue element, which is shallow, but only the deep line accumulates the red element, which is deep. It is also possible to install a tracer element like strontium at a given depth in addition to natural gradients. LEADER field tests across the U.S. show promise in detecting root depth differences in diverse maize germplasm. Molly has observed a very good relationship between actual root depth measured by soil cores and estimated root depth from LEADER, in this case R squared 0.76. Across multiple field sites, she is able to distinguish deep and shallow rooted maize lines. This is good news since root depth is very important for drought tolerance, nitrogen efficiency, and carbon sequestration, but is hard to measure with existing methods. So in summary, I talked about 3 phenes: multiseriate cortical sclerenchyma for stronger, deeper roots, high conductance xylem perforation plates for improved drought tolerance, and thicker parenchyma cell walls for reduced root cost and better soil exploration. I also talked about integrated phenotypes. I gave the example of the xylem architecture interaction for drought adaptation in Phaseolus species. I gave the example of clusters of integrated phenotypes explaining drought adaptation in maize. And I gave the example of how modeling rice for nitrogen efficiency shows the importance of synergism in plant performance. The importance of phene interactions creates a vast phenotypic morphospace we need to explore and understand. One solution I presented, evolutionary optimization. Another, emulation and statistical machine learning. I talked about phenotyping. I talked about shovelomics for field phenotyping of root architecture. I gave the example of how shovelomics led to the discovery of CypK for maize root growth angle. I talked about how that's evolving to a new generation, a pipeline called Root Robot Dirt 3D. I talked about anatomics for field phenotyping of root anatomy. I gave the example of the discovery of BHLH121 for root cortical larynxoma formation in maize to show that that platform works. That platform is being evolved to LAT 2.0 and RootScan 3D. I talked about LEADER for non-destructive estimation of root depth in the field. Obviously, this work represents the contributions of many people. This is the current configuration. Thank you very much.